Hello everyone. I am Docsaab and today I will be discussing vitamin B1 or thiamine, an essential water-soluble vitamin with critical roles in metabolism, neurological integrity, and cardiovascular function. In this presentation, I will take you through its biochemical significance, dietary requirements, deficiency syndromes, diagnostic methods, treatment strategies, and some important future research directions. To begin with, it is important to understand that thiamine is not a simply another dietary macronutrient. It is a core metabolic vitamin that supports fundamental cellular processes. Vitamin B1, also called thiamine or thiamine, is a water soluble and indispensable for normal human metabolism and cellular function. From a biochemical perspective, thiamine exists in several phosphorylated forms within the body. These include free thiamine. The non-phosphorylated form represents the basic thiamine molecule as consumed in the diet. It requires conversion to phosphorylated derivatives for biological activity. Thiamine monophosphate, present in cells at low concentration, approximately 6 to 7% in nervous tissue. Thiamine monophosphate is considered an intermediate in transformation pathways and biologically inactive on its own. Thiamine diphosphate, also known as thiamine pyrophosphate, this is the primary biologically active form of thiamine and the most abundant phosphorylated derivative in tissues. TDP function as an essential coenzyme for multiple enzyme complexes involving energy metabolism. Thiamine triphosphate TTP participates in non-coenzymatic reactions, particularly in membrane conductance and cellular signaling pathways. It plays specialized roles in nerve impulse transmission and cellular stress responses. And adenosine triphosphathiamine, this recently discovered adenyl derivative of thiamine triphosphate accumulates under cellular stress conditions, particularly during carbon or nitrogen deficiency. Before moving into modern clinical practice, it is worth appreciating the historical importance of thiamine. This slide presents a timeline of the discovery of vitamin B1. The disease beriberi, now known to result from thiamine deficiency, was long known as kake in Japan and China and was initially attributed to miasmas from wet soil or infectious organisms. Systematic scientific investigation began in the 1880s when Dutch researchers Cornelis Pekelhering and Cornelis Winkler studied beriberi in the Dutch East Indies, initially believing they had an isolated micrococcus responsible for the disease. The breakthrough came when Christian Ekman observed that chickens fed white polished rice developed leg paralysis or polyneuritis, while chickens fed brown unpolished rice remain healthy. This pivotal observation led to the recognition that a nutritional factor rather than an infectious agent was responsible for beriberi. Then Gerrit Grigens, who succeeded Ekman in Java, correctly concluded that unknown substances in foods were essential for peripheral nervous system function. In 1910, Japanese researchers Umetaro Suzuki, considered the father of vitamin research in Japan, identified aberic acid, making him among the earliest to identify a vitamin. In 1912, Polish biochemist Casimir Funk coined the term vitamin A, later shortened to vitamin, for what he believed was the anti-beriberi factor. The definitive isolation came in 1926 when Baron Jensen and William Donath successfully isolated and crystallized the substance that cured polyneuritis in pigeons. The final milestone occurred in 1936 when Robert Williams successfully synthesized thiamine, enabling its mass production. Let us now move from history to biochemistry. This slide introduces the major enzyme systems that require thiamine diphosphate, and as shown in the diagram, TPP is required at several crucial metabolic junctions, including pyruvate dehydrogenase. This multi-enzyme complex catalyzes the critical step of converting pyruvate to acetyl-CoA, effectively linking glycolysis to the citric acid cycle. This reaction is absolutely essential for cellular energy production from carbohydrates. Then alpha ketoglutarate dehydrogenase, operating within the citric acid cycle, this enzyme complex converts alpha ketoglutarate to succinyl CoA, generating NADH in the process. This step is crucial for continuous energy production through oxidative metabolism in the mitochondria. Then branch chain keto acid dehydrogenase. This enzyme complex serve as the rate limiting step in the catabolism of branch chain amino acids. These amino acids are essential nutrients and their proper metabolism supports protein synthesis, energy production, and neurotransmitter synthesis. Then 2-oxoadipate dehydrogenase involved in lysine catabolism. This enzyme converts L-lysine derived 2-oxoadipate to glutaryl-CoA and subsequently to acetyl-CoA. And lastly, transketolase functioning in the pentose phosphate pathway within the cytosol. Transketolase catalyzes critical carbon transfer reactions. This pathway produces ribose 5-phosphate, essential for DNA and RNA synthesis, and generates NADPH, which is required for fatty acid and steroid synthesis, as well as antioxidant defense through glutathione reduction. So, when we speak about vitamin B1, clinically we are really speaking about a vitamin that sits at the crossroads of ATP generations, redox balance, biosynthesis, and neuronal metabolism. Now, let's see metabolic pathways involving thiamine. From a metabolic standpoint, thiamine plays a central role in carbohydrate metabolism. It supports the pyruvate dehydrogenase complex, which links glycolysis to aerobic respiration, and transketolase, which links glycolysis to the pentose phosphate pathway. This integration allows efficient extraction of energy from glucose while also supporting biosynthetic reactions and cellular homeostasis. The second major point is energy production. TDP is indispensable for mitochondrial oxidative metabolism and therefore ATP generation. Through TDP dependent enzymes, thiamine connects carbohydrate, amino acid, and lipid metabolism to the electron transport chain. In addition to carbohydrate metabolism, thiamine also contributes to lipid metabolism. Through its role in acetyl CoA generation, it supports both fatty acid oxidation and the biosynthesis of isoprenoids and steroids. The pentose phosphate pathway, which depends on transketolase activity, also generate NADPH. That reducing power is essential for fatty acid synthesis. Another highly important area is neurotransmitter synthesis. TDP dependent metabolism contributes to the production of glutamate and GABA, which are central neurotransmitter in the nervous system. Thiamine also influences synthesis of acetylcholine, serotonin, and aspartate. This is one reason thiamine deficiency can produce such profound neuropsychiatric and neurological manifestations. Beyond classic coenzyme roles, it is also highlights thiamine derivatives such as TTP and ATP, which appears to accumulate during nutrient deficiency and cellular stress. That suggests thiamine may also play a role in cellular adaptation and metabolic signaling, not just in enzyme catalysis. As more advanced level, thiamine also appears to have non-coenzymatic functions. This is a particularly interesting area because it broadens our understanding of vitamin B1 beyond the classic enzyme complex model. For example, thiamine triphosphate or TTP may influence membrane chloride channels, thereby affecting neuronal excitability and signal transmission. Thiamine also implicated in nerve impulse propagation and myelin sheath maintenance, although some of these mechanisms remain under active investigation. It is also points to evolutionary conservation, noting that in plants and microorganisms, thiamine derivatives regulate gene expression through riboswitches. That is a useful reminder that thiamine biology is broader and more sophisticated than simple vitamin replacement tables might suggest. Let us now explore metabolism of thiamine. As showing in this figure, thiamine absorption occurs primarily in the small intestine through two distinct mechanisms depending on the concentration. At physiological or nutritional doses, absorption occurs via active transport mediated by specific thiamine transporters. Thiamine transporter 1 encoded by SLC19A2 and thiamine transporter 2 encoded by SLC19A3. This process is pH dependent and sodium dependent, functionally optimally in the jejunum. At pharmacological doses, means high supplementation levels, passive diffusion becomes the dominant absorption mechanism. Most dietary thiamine exists in the phosphorylated forms, which must be hydrolyzed by intestine phosphatases to free thiamine before absorption can occur. Once absorbed, thiamine enters the bloodstream and is distributed to tissues throughout the body. Once absorbed and transported in the bloodstream to tissues, thiamine is converted intracellularly by thiamine pyrophosphokinase into thiamine pyrophosphate, the active coenzyme form. Then thiamine is distributed throughout the body tissues with the highest concentration found in skeletal muscle, heart, liver, kidneys and brain. Approximately 80% of thiamine in whole blood is contained within red blood cells. However, human thiamine stores are extremely limited, typically only 25 to 30 mg in total. This limited storage capacity combined with thiamine's short biological half-life means that body stores can be depleted within approximately 2 to 3 weeks of inadequate intake. As a water-soluble vitamin, excess thiamine is readily excreted in urine. When intake exceeds physiological needs, thiamine and its metabolites are efficiently eliminated by the kidneys. This rapid excretion mechanism prevents accumulation but also necessitates regular dietary intake to maintain adequate levels. Now turning to nutritional requirements, the recommended daily intake of thiamine varies by age, sex and physiological status. For infants, adequate intake is 0.2 milligrams per day from birth to 6 months and zero point 0.3 milligrams per day from 7 to 12 months. For children, the requirement rises progressively from zero point 0.5 milligrams at 1 to 3 years to zero point 0.6 milligrams at 4 to 8 years and zero point 0.9 milligrams at 9 to 13 years. Among adolescents aged 14 to 18 years, males require 1.2 milligrams per day, whereas females require 1 milligrams per day. In adulthood, the RDA is 1.2 milligrams daily for males and 1.1 milligrams daily for females. During pregnancy and lactation, the requirement increases to 1.4 milligrams per day. In older adults, the same adult values are generally maintained. The key point is not just memorizing the numbers, but recognizing that thiamine requirement increases in conditions of higher metabolic turnover, increased carbohydrate load, pregnancy, lactation, systemic illness, and recovery states. Let's see about food sources and dietary intake. From a dietary perspective, thiamine intake depends strongly on food pattern and food processing. Foods with minimal or negligible thiamine contribution include examples such as cheddar cheese, roasted chicken, and sliced apple. By contrast, foods that provide approximately 0.1 to zero point 0.2 milligrams per serving include bluefin tuna, whole wheat macaroni, baked acorn squash, brown rice, enriched white rice, whole wheat bread, orange juice from concentrate, sunflower seeds, braised beef, plain low fat yogurt, oatmeal, boiled sweet corn, 2% milk, and pearly barley. Then let's see excellent dietary sources of thiamine defined here as food providing zero point 0.3 milligrams or more per serving. Examples include fortified breakfast cereals, enriched egg noodles, boiled pork chop, cooked trout, boiled black beans, plain enriched English muffins, and cooked mussels. This is clinically relevant because patients may assume they are eating adequately, yet actual thiamine intake may still be borderline depending on the refinement of grains, dietary restrictions, and illness-related poor intake. Now let's talk about thiamine deficiency conditions and clinical manifestations. Clinically, early thiamine deficiency can be difficult to detect because the initial manifestations are often non-specific. Patient may present with loss of appetite, fatigue, generalized weakness, irritability, and mood changes. They may also report difficulty with short-term memory and concentration, sleep disturbance, muscle weakness, and abdominal discomfort. The reason these symptoms are so important is that they often precede the classical syndromes of Beriberi or Wernicke encephalopathy. In practice, failure to recognize these early warning signs may delay diagnosis until much more serious neurological or cardiovascular damage has already developed. Now let's see types of thiamine deficiency diseases. At this stage, the presentation moves into the classic clinical syndromes of thiamine deficiency. First, dry beriberi primarily affect the peripheral nervous system. It presents with symmetrical peripheral neuropathy involving both motor and sensory deficit, diminished reflexes, sensory loss, and paresthesias such as tingling or numbness. The pathophysiology involves myelin loss in peripheral nerves, but notably without acute inflammation. Second, infantile beriberi occurs in breastfed infants or mothers who are thiamine deficient. These may present acutely with cardiac failure or more gradually with failure to thrive, vomiting, and neurological symptoms. Third, wet beriberi represents predominant cardiovascular involvement. This is medical emergency characterized by high output heart failure, tachycardia, peripheral edema, fluid retention, dilated cardiomyopathy, and sometimes chest pain. The underlying mechanism involves impaired myocardial energy metabolism along with dysautonomia. One of the most clinically urgent manifestation of thiamine deficiency is Wernicke encephalopathy, an acute neurological syndrome caused by severe deficiency affecting the brain. The classic triad includes ocular abnormalities such as nystagmus or ophthalmoplegia, confusion or altered mental status, and ataxia or gait disturbance. Additional features may include nausea, vomiting, fever, and progressive cognitive impairment. Although it is commonly associated with chronic alcoholism, it can occur in any severe deficiency state. Korsakoff syndrome represents the chronic phase of deficiency related brain injury and is marked by severe memory impairment, especially of short term memory, psychosis, and confabulation, in which the patient unintentionally fabricates memories to fill in gaps. Together, these conditions form Wernicke-Korsakoff syndrome, a continuum from acute to chronic deficiency-related neurological damage. The key clinical point is that delayed treatment may result in permanent neurological disability, so thiamine must be given promptly when suspicion exists. Beyond nutritional deficiency, thiamine biology is also relevant in genetic disease. This slide highlights thiamine responsive megaloblastic anemia syndrome or TRMA, also called Rogers syndrome. This disorder results from homozygous or compound heterozygous mutation in SLC19A2, the gene encoding thiamine transporter 1. The classical triad consists of megaloblastic anemia, early onset diabetes mellitus, and sensorineural deafness. However, the phenotype may also include cardiac arrhythmias, structural heart disease, optic atrophy, cone rod dystrophy, seizures, thyroid dysfunction, and other neurological features. The slide also references more recent evidence showing that heterozygous loss of function mutation in SLC19A2 may produce an autosomal dominant diabetes phenotype with mild TRMA features. This is mechanistically linked to impaired thiamine uptake, mitochondrial dysfunction in pancreatic beta cells, reduced insulin secretion, early onset diabetes, decreased cell proliferation, and greater susceptibility to oxidative stress. This is a reminder that thiamine related pathology can arise from both environmental deficiency and inherited transport defects. To understand the clinical manifestation of deficiency, it is useful to summarize the pathophysiology in a structured way. First, there is metabolic failure. Without adequate TDP, cells cannot efficiently derive energy from glucose through glycolysis linked mitochondrial metabolism and the citric acid cycle. This is especially damaging in organs with high energy demands, particularly the brain, heart, and peripheral nerves. Second, there is lactate accumulation. If pyruvate dehydrogenase activity is impaired, pyruvate is shunted toward lactate production, which can contribute to lactic acidosis and further cellular dysfunction. Third, there is cardiovascular compromise. The myocardium requires a constant high energy supply, so impaired energy metabolism can lead to dilated cardiomyopathy, high output failure, and autonomic dysfunction. Finally, there is neurological damage. The brain depends heavily on glucose metabolism, and energy depletion disrupts neurotransmitter synthesis, neuronal function, and myelin integrity. That combination explains the neurological and neuropsychiatric spectrum of thiamine deficiency. In essence, the pathophysiology is unified by one central concept: energy deprivation in metabolically active tissues. Now let's talk about assessment and diagnostic methods. For direct assessment method, when assessing thiamine status, specimen selection is critical. The whole blood is the preferred specimen because approximately 80% of blood thiamine is located within the red blood cells. That point matters because plasma thiamine reflects more recent intake and does not reliably represent tissue stores or functional status. Then method described here, liquid chromatography, tandem mass spectrometry, is considered the most sensitive specific, and precise way to measure thiamine diphosphate in whole blood. Because it directly measures the active coenzyme form, it gives the most accurate insight into clinically relevant thiamine status. Then, this slide presents additional biochemical approaches to thiamine assessment. High performance liquid chromatography with fluorometric detection is commonly used in clinical samples including whole blood, serum, urine, and breast milk. In this method, thiamine is converted to thiochrome, which fluoresces under ultraviolet light and allows sensitive detection. The slide also discusses urinary thiamine excretion, which can reflect a recent intake and can help identify possible deficiency. Urinary excretion below approximately 27 micrograms per day suggests inadequate intake or impaired absorption. However, interpretation must be cautious because urinary measurements are influenced by hydration status and renal function. So, although urinary testing can be useful, it is generally best interpreted in the broader clinical context rather than in isolation. A particularly valuable functional assessment method is the erythrocyte transketolase activity coefficient. This test measures transketolase activity in red blood cells before and after addition of excess thiamine. The resulting ratio reflects the degree to which enzyme activity improves with supplementation. Values greater than 1.25 are indicative of thiamine deficiency, and higher values suggest more severe deficiency. What makes this test especially meaningful is that it assesses functional adequacy at the cellular level rather than only measuring concentration. For clinicians, that distinction is important because functional deficiency can sometimes exist even when simple concentration based interpretation is unclear. Let's look at the treatment of thiamine deficiency. Management of acute deficiency must be prompt, especially when cardiovascular or neurological symptoms are present. The initial regimen is 200 milligrams 3 times daily, given either intravenously or orally, and continue until symptoms resolve or improvement plateaus. After this acute phase, maintenance therapy consists of 10 milligrams per day orally until expected recovery is complete. An alternative acute regimen listed here is 50 milligrams intramuscularly for 2 to 4 days, followed by oral maintenance therapy. The larger clinical principle is simple. When deficiency is suspected in symptomatic patient, especially with neurological risk, treatment should not be delayed while waiting for confirmative testing. In suspected Wernicke-Korsakoff syndrome, treatment needs to be significantly more aggressive. The initial phase, in which the patient receives 500 milligrams intravenously 3 times daily on days 1 and 2, infused over about 30 minutes. This is followed by secondary phase of 250 milligrams daily, given intravenously or intramuscularly from days 3 to 5. The World Health Organization recommendation for pregnant women with beriberi 10 milligrams orally each day for 1 week, followed by 3 to 5 milligrams daily for at least 6 weeks. This slide highlights a very encouraging but important point. When thiamine deficiency is treated early, patient often improves substantially. In Wernicke encephalopathy, ocular signs, gait abnormalities, and some language-related problems may resolve. Cardiovascular manifestation can improve dramatically within hours to days, which is one reason prompt diagnosis and treatment can be so rewarding clinically. However, the degree of neurological recovery depends heavily on how long the deficiency has been present, how severe it became before treatment started. In particular, Korsakoff syndrome may leave persistent deficit in memory and cognition even after treatment. So, while thiamine replacement can be highly effective, the timing of intervention remains critical. Let's move beyond classical deficiency and explore new areas of research involving thiamine. One area is cancer metabolism. A 2025 study examining the relationship between vitamin B1 intake and risk of non-melanoma skin cancer in US adults. Since thiamine pyrophosphate influences cellular energy generation and proliferation, there is growing interest in how thiamine availability may affect tumor biology. Second area involves fermented foods and microorganisms. A 2025 systematic review cited here examined vitamins formed during fermentation and their effects on human vitamin status. This is especially interesting because beneficial bacteria and yeast can increase thiamine content in foods, potentially improving nutritional quality. There is an ongoing clinical trial in multiple sclerosis evaluating combined thiamine and biotin therapy. The therapeutic hypothesis is that such supplementation may be safe, well tolerated, and potentially supportive of myelin maintenance and neurological function. The final research slide broadens the scope even further. First, in parasitic disease research, scientists are studying thiamine metabolism in Plasmodium, the malaria parasite. Although the parasite can synthesize thiamine, its thiamine dependent pathways may still reveal vulnerabilities that can be exploited therapeutically. Second, there is interest in skincare and cosmeceutical science, where thiamine is being explored for possible antioxidant, anti inflammatory, and cellular energy supporting effects in skin tissue. Third, a 2024 retrospective and marketing analysis concerning use of B vitamins, including thiamine, in the pharmacotherapy of tuberculosis. Given the metabolic burden of chronic infection and prolonged treatment, the role of micronutrient support remains clinically relevant. As we come to the end of this presentation, I would like to summarize the central message. Thiamine is a small molecule with major clinical importance. It is essential for energy metabolism, neurotransmission, myocardial performance, and neurological stability. Deficiency can present subtly at first, but if missed, it can progress to severe and potentially irreversible disease. Thank you very much for your attention, guys. I welcome your feedback and will be happy to discuss any questions. If you found this presentation helpful, please like, share, and subscribe to the Docsaab Diary for more educational content.